Friday Live Classroom. We've been talking about cell division all week, and today we are going to talk about a specific segment of cell division, which is mitosis. So I'm going to be using some everyday objects to show you exactly how it is that mitosis occurs. Okay, I want you to imagine that this plate is a cell. And we're not going to talk about all the organelles today, just the organelles that are involved in cell division. So a cell contains a nucleus. So this stands for the nuclear membrane. And inside the nucleus, we have DNA. And now this is during G1 of the cell cycle, so the DNA is in no particular order. It's not condensed into chromosomes, it's just sort of floating around in the cytoplasm and kind of a mess. It's chromatin right now. So this DNA is not currently organized, it's chromatin just kind of in here every which way. And the other organelle that's involved in mitosis is the centrioles. So centrioles always occur in pairs and they hang out next to the nucleus. So right now, this is G1. This is where the cell spends most of its life. You have the nucleus, you have chromatin, and you have the centrioles. Now, we are still in interphase, but now we're at the second phase of interphase, which is S phase. During S phase, the DNA replicates. By replicating, we mean doubles. The DNA makes a copy of itself. So now we have 2 strands of each color because you're going to need 1 copy to go into each of the new cells. So this is still chromatin. It is still just sort of messy, unorganized, floating around in the nucleus. It's unwound so that the cell can make copies of the DNA that it needs to in order to make proteins. So this is S phase. The cell made a copy of all of its DNA. Now we're going to move on to G2. Now, during G2, the cell grows a little bit more and it replicates some of its organelles. Now, we don't have very many organelles to show, but the organelle I can show that it's definitely going to replicate is the centrioles. So it's going to need a second set of centrioles in order to perform cell division and so that each new cell gets a pair of centrioles. So during G2, the cell grows more, it's preparing for division, and it's adding that extra set of centrioles so that it can divide. Now we're going to move on to the mitotic phase. In the mitotic phase, we have mitosis and cytokinesis. Mitosis is composed of 4 stages: prophase, metaphase, anaphase, and telophase. So right now, we're going to do prophase. During prophase, a lot of things happen. So the very first thing that's going to happen is the nuclear membrane, so that border around the nucleus is going to dissolve. It's going to go away. So we're going to set that to the side. Another thing that's going to happen is the centrioles are going to move to opposite ends of the cell because they're the things that are going to be making the spindle that's going to direct everything. So you need 1 on each side so that they can pull the chromosomes to either side. And then the chromatin is going to condense and it's going to form chromosomes. So that's what I'm doing with the beads right now. I'm organizing them. They are now organized into chromosomes. Made them X-shaped because most chromosomes have an X shape. This is prophase. During metaphase of mitosis, remember M for middle. So these chromosomes are going to line up along the middle of the cell. So we're going to line them up along the middle. And when we say middle, it's going to be making like an equator. Think of the centrioles as the north and south pole, and then these are the equator going down the middle. So it's almost like a division sign. So this is metaphase. They're lining up down the middle. During anaphase, remember A for apart. So these sister chromatids are going to pull apart. So one chromatid is going to go toward one set of centrioles, one chromatid is going to go towards the other set. Same thing with the red, one's going to go toward each end, and same thing with the green. They pulled apart. A for apart, A for anaphase. In telophase, remember T for 2. There are now going to be 2 nuclei. So a new nucleus is going to form around this set of DNA, and a new nucleus is going to form around this set of DNA. And the DNA is also going to relax and unwind back into chromatin again, so it's no longer organized during telophase. At this point, mitosis is complete. Mitosis is only the division of the nucleus. The cell itself hasn't divided yet, but the nucleus has divided. Mitosis is only the division of the nucleus. So mitosis is complete now. However, cell division is not complete. There's one more step left. So, as I said before, mitosis is now complete, but there's one more step left in cell division, and that is the division of the cytoplasm. So cytokinesis is going to occur. So the cytoplasm is actually going to separate and divide into 2. And half of your DNA is going to go into one cell, and half of your DNA is going to go into the other cell. So you now have 2 cells that have DNA that's identical to the cell that started. These are your daughter cells. Their DNA is identical to the DNA of the parent cell. Now the cell cycle is complete, and each one of these daughter cells is now going to go through the cell cycle herself. Thank you for joining me for this journey through mitosis. I really hope that my demonstration has helped you to understand how mitosis works a little bit better. Next week, we'll also be working with the bees, but we will be performing meiosis, which is the production of sperm and eggs. Hope you enjoyed this, and I will see you in class. Have a great day.